I'm going to hand it over to Sanjay now and he's going to host the next session.
Thank you all.
Thank you.
Hello, good morning, everybody. I am Sanjay Malunchkar. I'm a software engineering manager in Engineering Services Group. I hope I got the name right, in Somali's organization. As Todd alluded to earlier, I'm one of those people who have been fortunate to be involved with STARR since its prior incarnation for almost 2 decades. It makes me sound really old, but STARR has come a long way during that time. STARR went from just the EMR data warehouse of both hospitals to now include radiology, vitals, EKG, echo, and several other ancillary data sets. This morning, we are very fortunate to have distinguished speakers who are going to walk us through their research use cases. They'll be covering various aspects of STARR, such as chart review, bedside monitoring, real-time research, alerts, and much more. Without further ado, I would like to introduce our first speaker, Monica Granucci. Monica is a project management specialist with neurosurgery in the Cancer Clinical Trials Office. She uses STARR for retrospective chart reviews in neurosurgery and neuro-oncology, and to monitor disease incidence and patient flow rates in the cancer center's clinic. Before I hand it over to Monica, I would like to remind that please use Q&A to raise your questions. Monica, take it away, please.
Thank you for that introduction, Sanjay. My talk is going to be distinguishing seed from soil, a MetNet group use of STARR. And I just want to say first that I'm happy to go to be the very first presenter, because that means I get to make outer space jokes about STARR, and it looks like I thought of it first. So the title of my talk, Seed from Soil, comes from a little advance, an 1889 surgeon named Stephen Paget, who hypothesized, he was thinking about cancer metastases and noticed that his breast cancer patients had metastases in certain numbers of organs, certain types of organs preferentially. And he wanted to ask the question, why are cancer metastases more common in some specific organs than others? In particular, breast, lung, melanoma, and renal cancers all seem to like to metastasize to the brain for some reason. It's much more common than moving to other areas. And Paget came up with the idea that when a plant goes to seed, the quote from his famous presentation is, its seeds are carried in all directions, but they can only live and grow if they fall on congenial soil. So a cancer that arises at a primary site then sheds these cells, these seeds, into the body, and they travel everywhere, but they only take root, pun intended, thank you, Steve, better in some organs over others, different soils, different microenvironments that are welcoming to the tumor in some way. So then the question, of course, becomes, how can we shore up the brain's defenses so that cancer never takes root? And if it does take root, how do we make sure that we can kill every last cancer cell without disrupting the normal function of the host organ of this metastasis? So where to begin unpacking all of this? The government threw some money at it, and thank you for that, because they are paying part of my salary. The NIH's Metastasis Research Network, which is abbreviated the MetNet, uses systems-level approaches to understand cancer metastases by supporting collaborative multidisciplinary projects that utilize systems-level approaches. Stanford is one of them, and when we received funding, we have a group of more than 12 PIs and all of their labs that work together to address this in the context of brain cancer. Our specialties are from all over. You can see some examples on the screen. I won't go through them all, but we're all working under a core set of guiding principles. we are trying to understand and disrupt the cancer neuroimmune axis supporting brain metastases. It's particularly complex in the brain specifically. We know so little about it compared to other organs because it's all locked up in your head, and you don't take margins in brain cancer. You can't take extra tissue. You know, if there's a tumor in someone's motor cortex, you can't make sure to cut everything around the cancer as well as just the cancer itself to make sure you get everything, because the person can't walk anymore. So we don't have a lot of extra tissue to play with. We don't have a lot of extra space to research. We only get very, very small samples, and you don't take normal brain. It stays where it is. To add to that, the immune system works differently in the brain. Some people call it immune privilege. We're understanding as we learn more that that's not quite an accurate assessment, but the immune system is different in the brain from most other places in the body. And so it's different in how it kills protocancer and cancer cells. Very little is known just at the baseline about the distinct mechanisms that drive tumor cells to the brain and allow them to grow. But we do have evidence to suggest that one way they do grow in the brain is they hijack normal brain cells called resident microglia. Now, microglia are these cells that support neurons and make sure that they're growing well. And what the cancer does in the brain is it hijacks those microglia and gets them to support it instead. So our MetNet Center's focus is on the tumor cells and what makes it, what about the microniche that they find is attractive, the resident microglia and how they are hijacked, and what is different about the immune system and how it's acting in all of these ways. Now there's a whole lot that I could get into. It is, involves 12 labs across the campus, but I want to focus specifically on the work that I've done with the neuropathology core supporting all of this research and what we've done with STARR. The researchers that we initially sat down with were limited in the scope of their research by how much they could collect from scratch from EPIC, because the first step is always to identify a large enough sample size to be able to get significant results. And then the issue was that once a researcher on their own built this cohort painstakingly, they only had so much time remaining to collect and analyze data before they ran out of funding. And the scope of projects was being constrained by this. So we were asked as part of this larger initiative to lower the barrier to brain met research by providing smaller research groups who were looking to make an impactful go of things with a large patient pool and a platform with which to perform the research. So of course, we thought of STARR, and we thought of STARR as the data lake. And this allows for more time to be spent on a deeper dive into a topic than each individual group can manage on themselves. So what we did is we first turned to your lovely cohort discovery tool, and we pulled patients with a diagnosis of brain metastases at any time point who had visited Stanford within a 10-year timeframe from 2008 to 2018. And we relied initially on the word for it. Oh my gosh, I'm forgetting the name of the tool. At this step of the process, we collected more false matches than true to ensure that we collected everyone. So we relied on algorithms to initially cast a wide net, and then we narrowed our search with individual eyes-on charts by humans. One wrinkle we had to contend with was that the 2008 to 2018 time period included the switch from ICD-9 coding to ICD-10 coding, and we were using ICD-9 coding for the brain metastasis diagnosis as the way to pull these charts. But once we had accounted for this, we cast our initial wide net. That's right, the chart review tool. And then again, like I said, we reviewed each chart, person by person, chart by chart, and removed anyone who did not have documented evidence that we could find of a brain metastasis in their chart, instead of just relying again on ICD-10 codes. So this double confirmation system. We ended up with about 5,000 charts. We were left with a smaller subset of patients with confirmed brain metastases who then received care at Stanford over this period. And since 2018, we've updated it every year for the last 4 years. And we have a significant data set now built up for researchers to be able to go in. Let's say that I have a researcher interested in GI cancers and how they spread to the brain. we have over 400 people over the last 12 years that have this diagnosis that we can turn to immediately and say, you start your look here. Instead of starting with the entire data lake, start here. The indexing that we do isn't overly specific. For instance, patients with a salivary gland cancer or a primary squamous cell carcinoma of the tongue, they're all sort of under the umbrella of an oropharyngeal cancer. So we wanted to give people sort of buckets in which to pull from, but it's specific enough to be a starting point. And over the last several years, we've worked with over a dozen research groups, but I want to tie this back to the MetNet research that we've been doing. The MetNet Neuropathology Core used these initial listings to then locate available paraffin-embedded tissue that was resected from patients who underwent surgery here at Stanford to identify tissue for a microarray construction. Now, what is a tissue microarray? Microarray, it's a little block of wax with up to 1,000 or more tissue samples embedded into it. And then you wash, the important thing is you wash this tissue with anything that sticks to it. You can look for DNA markers, you can look for RNA markers, you can look for protein markers, but it's this really flexible tool that allows you to do a high throughput analysis of multiple specimens all at the same time in identical standardized conditions on a single slide. And then because you can take this slide and then directly image it, it provides a really fast turnaround time, limited maximal use of archival tissue samples because these samples are very, very small. So just taking a single shaving from a single slide to be able to do this imaging is really useful. And then also this core can be used over and over again. We are very lucky. The commercial TMAs, that tissue microarrays that we were working with just weren't cutting it. The best commercial TMA we were able to find was only 43 cases of anyone with brain metastases, and only 5 of those people had breast cancer. Versus the TMA that we were able to construct from Stanford resources has 254 samples from 150 patients with 6 different tissue types. And that includes at least 6 patients that I know of, I wasn't directly involved in the TMA construction, who have samples present on the TMA, both from their initial primary cancer of the breast and from tissue taken from their brain metastasis from their initial breast cancer, so that we can tease out the differences between those 2 cell types and what might lead to one becoming the other. At this point, the MetNet group has a breast-to-brain TMA and a lung-to-brain TMA, and we're currently working towards building a glioblastoma TMA as well. Then what we plan to do with this microarray is we plan to use the generously donated CSF, cerebrospinal fluid, from patients who have come to Stanford with cancers that have metastasized to their brain and agree to donate. Then we take the CSF and we look at it, we spin it down, we extract cellular RNA, cell-free RNA, and we start looking for promising diagnostic and prognostic markers and targets, which we can then turn around really quickly and use the TMA to see if they're worth investigating, if it's just limited to this specific patient, or if it's part of a larger trend that we see across different people. So the TMA is really promising because it gives us a way to sink that additional like barrier, that resource intensive first step, and have it conserved across different groups so that the barrier to entry is much lower. So what we plan to do in the future is then tease what comes out from the tumor cells, which and what are hijacked from the resident microglia, and what comes from the immune system in response to this invasion, to see what we can learn in order to combat this specific, very difficult kind of cancer. And that brings us full circle on how we've been using STARR to do some really cool research in brain tissue. I would like to acknowledge all of the hard work that all these people have done. In particular, thank you, Melanie Hayden-Gephardt, for leading the MetNet here at Stanford, and thank you to Susan Weber as one of the biggest contributors to the STARR chart review tools. It's been an invaluable part of our research. Does anybody have any questions that they have for me at this point?
So, Monica, I have a question here. I'm not sure if you're involved with this particular research, prior to start using chart review. If you do, could you tell us what was your workflow before chart review came into picture and how it has hopefully improved your research?
Sure, absolutely. So previously, what researchers had been doing is relying on individual physicians to compose lists of patients they were interested in following for research years down the line, because it's such a huge lift to go and do that retrospectively. The physicians were stuck doing it prospectively, writing down, okay, this person came in, and they could be, I'll keep this list in an Excel file squirreled away on my personal computer. Maybe it will be useful in the future at some point. And then we would come to them later and say, do you have any patient lists of people with lung cancer that's metastasized to the brain? And it'd say, oh, well, I think I might. And we'd have to go to all these different physicians. And some of them had been keeping the list, some of them hadn't. Some of them were keeping lists that contained that, and you'd have to look through it. And it was just a really inefficient way of being able to look back through this. There's not actually a tag, there's not an ICD-10 or ICD-9 code to encode for the different types of brain metastases. It's reliant on primary cancer being coded and then brain metastases being tagged, but you can't rely on that code in that level of detail that we need, because oftentimes ICD-9 or ICD-10 codes are applied for things that clinicians are unsure of in a chart. We're looking to see if this is true, and that's why we are doing this MRI, or that is why we are taking this CT. And so it winds up in the chart even if a patient doesn't have a brain metastasis, for insurance purposes. So we're really grateful to STARR for being able to go through that incredible amount of data retrospectively and be able to make sense of it. Did that answer your question, Sanjay?
Yes, thank you. Thank you, Monica.
Great. Any other questions from the group? All right. Thank you, everyone, for allowing me to go first. I really appreciate your time.
Thank you so much, Monica, for that great presentation. I'm sure this will be an inspiration to others to start using chart review more and simplify their workflows and be smart about their research and have better outcomes. So without further ado, our next speaker is Ethan Steinberg. Ethan is a PhD student in the Department of Computer Science. Ethan will be talking to us on self-supervised time-to-event modeling with structured medical records. I know that sounds handful. I don't know what it means, but Ethan used STARR to build long-term risk models for various medical conditions.
Hello, good morning. Can you hear me and see my slides?
Yep.
Perfect. Yeah, so here we're going to be talking about, as mentioned, self-supervised time-to-event modeling with structured medical records. To start with, I assume most people know this, but what is an electronic health record? It's basically an ordered sequence of events of various types, all arranged on a timeline. Each patient has many of these things going on. The key insight for this work in particular is that we can view this structured data as sort of like a formal language of things. So like in our EHR, a sequence of symbols follow probabilistic patterns in a similar way like the English language might. So like quick jump, fox jumps over the lazy dog. In the EHR language, we have this like sequence of symbols which represent these medical events that are occurring to patients. And this sequence is informative. Like if you see someone with a diabetes diagnosis, you'd expect them to have various diabetes medications for in the future. And this key insight is that you can use over the years, machine learning has developed a lot of techniques for relying on the structured sequence of English language. And we can use some of the similar techniques working with our sort of EHR language. In particular, the concept of a foundation model and transfer learning. So we have this huge unlabeled data set of EHR records. We can use this to train a foundation model to sort of learn that internal structure, that internal sequence ordering, or the pattern within that internal sequence ordering. Then we can adapt that model to solve individual models within our electronic health records, individual like risk prediction tasks. And because we're adapting from this large model and you taking advantage of that internal structure, we can use a much smaller label data set for solving these sort of medical risk problems. So the focus of this talk is, can we create a sort of like a self-supervised foundation model for EHRs that specifically helps for timed event tasks? Basically, can we learn the intrinsic structure within our unlabeled EHR data sets in a way that helps us to train like various types of timed event models? Why timed event in particular? It's hard for existing techniques, mainly because it's a very high parameter space model. And it's also very important in clinical applications. Because basically in healthcare, we always have to, almost all of our tasks and setups have censoring. We never have a full patient's medical history. That medical history always gets either cut off when they either leave our health system or they leave the state, or we just lose track of them for whatever other reason. So the proposal of this work is to introduce MOTOR, which is many outcome time-oriented representations, to help solve this issue of both time event tasks and EHR data with the foundation model. For people who are unfamiliar, a time event model tries to predict the risk of a particular patient across time. So here's some simulated data showing a couple of patients, A, B, C, D, and their survival probability across time. Well, survival is the most common time event task, but in theory, you could do this with any particular objective, like say you're doing cancer risk across time. Maybe the same, you're just tracking those probabilities as they evolve over time. The main issue is it's very parameter intensive because you're fitting not only a model to predict like risk at a single point, but you're predicting risk at every possible time point. So there's sort of three components of MOTOR. First, we have to set up a pre-training task that we use to fit our large foundation model. We have to design that foundation model to learn that pre-training task, and we have to transfer it to individual target tasks. So for our pre-training task setup, we use the insight that there's a lot of various, we can use the timed event of all various IC codes within our data set to construct a bunch of timed event tasks. So for every single, or not, for the highest entropy codes within our data set, we construct a bunch of timed event tasks for every single patient in our data set. So we've created this huge labeled data set of millions of patients and also thousands of tasks, where we predict the timed event at every single time point in the patient history. Now we have to define a model architecture for learning that task. We follow prior work and mainly just use a pretty straightforward transformer that takes as input the medical record. So given the codes of the patient and also the timestamps for when those codes occur, we encode both of those using just regular embeddings and also positional embeddings for the timestamps and feed them into a relatively standard transformer. So given those two components, now we also need a way of defining our timed event model. We use what's called a piecewise exponential timed event model, where the main idea is you assume the hazard rate of each patient is sort of like a piecewise constant function. So you take the whole universe of time, you split it up into these little pieces, these little linear pieces, and you learn a single hazard for each of those little pieces. This model has a lot of advantages. One, is very flexible. In theory, this can fit, depending on how many pieces you pick, you can fit almost any timed event distribution. And second, it's very parameter efficient. It allows you to share parameters or share some of the pieces between each model, between each task within our large model, which is important because we're fitting 8,000 of these things. So we need to make it at least somewhat efficient. And also we use a log linear hazard. So the log of the hazard is log of the internal representations. So we can then take our model, so we fit all this thing. So we have this large transformer model that we fit. And now we can refit just the log linear layer at the bottom. So this final layer right here for individual tasks. There's slightly a tricky bit due to censoring. We have to use like a second order optimizer for this, but it's a linear model, so it is relatively straightforward to optimize within that setup. So given this, we evaluate on 6 tasks. So we predict celiac disease, heart attack, lupus, fatty liver disease, or non-alcoholic fatty liver disease, pancreatic cancer, and stroke. These 6 were selected because they're relatively important clinical tasks, and they also represent a mix of both sort of acute conditions and also more chronic conditions. We compare versus several other baselines on the C statistic. So how well can we actually rank patients in terms of risk? And we can see that we get pretty drastic improvements over generally the best baseline, which is random spiral forest. And we also do better. One important baseline here is motor WP, which is motor without pre-training. So we take the same architecture, our fancy neural network, and we don't do any pre-training whatsoever. So we don't do the fancy 8,000 pre-training tasks. And you can see that performs quite poorly because we simply do not have enough samples for these various tasks for that large model. You need the pre-training over the thousands of tasks to actually get the benefits you want. Some other side results is because we have this foundation model, we can actually subsample our task data sets. So we do like 5, 10%, 25% samples of our celiac, heart attack, lupus, non-alcoholic fatty liver disease, pancreatic cancer, and stroke data sets. And we're able to obtain pretty good models even with a very small number of samples. The reason why this is the case is because we're only fitting this tiny linear model, tiny 512 parameter linear model when we do the fine tuning. And it doesn't really take that much data. You only need a couple hundred examples to actually fit those 500 parameters, especially because that model itself is regularized. And so even with 5% of samples, I mean, you do have degradation, but even with 5%, you can get pretty strong models. A lot of people in my lab have contributed to this, or I guess my professor's lab, Nigam Shah's lab. Here's a list of various people. I also want to talk briefly about, there's a lot of benefits of STARR OMOP in particular for this research versus other formats of the data. Because it's an OMOP data source, we can write our code in a generic OMOP form and have all of our model take features in a generic OMOP concepts. And this allows our code and our models to actually generalize to other data sets, in particular TruVin and SickKids. TruVin is a large claims data set. SickKids is a Canadian hospital we're working with. TruVin, the transfer is not very good in terms of the code works on TruVin, but the model doesn't because the claims versus hospital is too large of a gap. We're actually able to see surprisingly good performance for transfer to SickKids. That's still mainly a work in progress. We're still fine tuning those results, but it is something you would not be able to do if this data was not in OMOP form because we wouldn't have everything normalized to the extent where the code and model can actually port. We also have our code base right there, GitHub Child with Femur, with tutorials for using this work. This is on archive currently, and we're planning on submitting this to NeurIPS in a couple weeks. That is it. Any particular questions?
So Ethan, I have a question. I noticed that one of your slides, you talk about using ICD-9 codes, ICD-10 codes, CPT codes, and also I think if I remember correctly, RX norm codes.
Mm-hmm.
Was there any particular reason why you used like a mix of different terminologies?
We generally just use whatever the raw data source has. Although note that those internally, could we take advantage of OMOP tooling? So we map all those internally to OMOP standard concepts. But we just use whatever codes are available in our source data.
Okay. Okay. Thank you.
And that's what's available within STARR OMOP. So that's what we use.
I see. I see that STARR OMOP is really helping your research with, by the nature of OMOP being a common data model, which allows you to run, I guess, federated queries with, different collaborators outside of Stanford.
Mm-hmm. Well, and also TruVin, which was in Stanford, but it's still within OMOP, so makes things more standard.
Okay.
Sorry. Well, did you have a second part of that question? Sorry.
No, I believe Aditya has a question.
Hi, good morning. Thank you for this. This is super cool. Just had a question with the way you're doing tokenization. Does
Mhm.
order matter when you're tokenizing an object at time? So thinking about like language model, specifically am I versus I am, there's a huge difference in language. Is it going to matter if your collection of ICD codes is in different ordering?
That's sort of an interesting question. So to some extent, it doesn't matter because we're explicitly providing the timestamps. We do require things be ordered causally in time because we prevent linkages. We prevent linkages across. We prevent linkages from the past to the future. We prevent information flow from the past to the future. But in general, the order. I mean, the model is order sensitive, but it's trained with a random order each time. The order only really matters for events that have the same timestamp. And unfortunately, the model does incorrectly use that order, like not use it in a like technical sense, but like the model does depend on that order, as in if you swap the order, results will change slightly. But I don't think the results depend too much on that because we train with random shuffled orders, and I don't think it has too much of an effect.
Yeah, I was just curious if you did any analysis on that, because like theoretically it's full cross attention across all of the tokens. So hopefully it gets kind of white noised out, but I was just curious.
Yeah, I mean, the way to think about this in Mental is like when you construct a transformer, you explicitly construct like this attention map that like defines the attention tokens it looks at. Clearly, that attention map is order dependent because we do a causal mask. So a few tokens can only attend on prior tokens. In theory, you could fix that. You can make that token mask actually like order independent, or you can make that attention mask order independent. But that's sort of complicated because that requires actually changing a bunch of like CUDA code that implements these like transformer kernels and stuff like that.
No, I understand that. I was just implicitly thinking you're doing like a Roberta going left to right, and then just masking the next. We can
Oh yeah,
take
that
this
is an important
offline.
point. We are using a causal attention mask for our transformer, so information only flows from the past, only flows from the past to the future.
Thank you. We can take the rest of the technical stuff. I'll ping you. Thank you.
If you have particular questions, you can see our code base, which has the various. I mean, it's just a standard causal attention mask. We also did. I just want to say we also did some experiments just to make sure the position encodings were important. If you move these position encodings, you do lose performance, although not as much as you might think.
Thank you, Ethan. I also had a follow-up question. I just wanted to give others a chance to speak. Would you be able to discuss your technology stack as to whether you're using Nero or, how did you or where did you perform your research?
We do all these experiments on Nero and Carina. Carina is sort of like a new cluster that's being spun up. One important note is we do not use BigQuery. We download all the files from BigQuery onto the sources so we can process things locally. Because when you're trying to feed in like the entire medical data set into like a transformer model, you really do want those files locally so you can actually load them at a reasonable speed.
So were they like CSV files or what kind of?
Yeah, we download the CSV files. BigQuery has a export to CSV option.
Okay. Thank you.
Yeah, so I guess we are using BigQuery, but just to download, not to actually do any parts of the analysis.
And do you remember how big your data set was? Like in terms of records, gigabytes, terabytes, or number of patients?
That gets into the Stanford raw data is about. Those are in the paper. I don't have them offhand. I know Stanford compressed. When you actually compress all of Stanford's data and you focus only on the coded data, it's only actually about 8 gigabytes. But the raw download is about 2 terabytes because most of it's like text information that I discard. Yeah, and for reference, Truven's about 20 gigabytes. Truven's about 2.5 times bigger than Stanford's data. Although it's much different because Truven's like a lot of more patients, but less data per patient.
Yeah, can you elaborate on Truven? Is that did you say it's a Stanford data set?
Yeah, or at least not Stanford. We have license to it through PHS.
Okay.
I think it's a university like license. I think it was a per like user fee. I think I'm not sure the exact setup, but
okay. And that data set has EMR.
That's
Yeah.
a claims data set.
Oh, claims data set. Okay.
So did you guys convert that data into OMAP? Is the proven data set in OMAP or,
Yes.
okay,
Yeah, and we also have some results on that, which I did not include in here because I think people are more interested in STARR. But you get similar, well, much less benefit with Truven because the whole point of this is transfer learning. At a certain point, you have enough samples in the data set itself that you don't need this sort of transfer learning. Once you get like 100,000 celiac patients, like there's really nothing else to be learned. At a certain point, you just have the max amount you can possibly learn on that particular type of patient.
Also, would you be able to tell us how easy it was to work with STARR OMAP dataset?
It's more difficult than Truven because some parts of it are there's a lot of remaining issues and bugs that we're still in the process of resolving. I think we've reached out to you guys about some of those issues. The most important one is a lot of the timestamps in STARR OMAP are wrong. We had to fix post-process fix.
Okay.
As in, like, they don't actually represent the sequence of medical records. And so if you don't fix the timestamps, you get like 100% performance because you have future information leaking to the past. Well, not 100%. You get higher performance than you should get.
Right.
Our repository actually has a bunch of those fixes in there. We have a whole file called like fixing STARR OMAP or whatever that tries to employ most of those fixes.
Great. If there are no further questions, thank you so much, Ethan, for the wonderful presentation.
No problem. Thank you for your work on this data set infrastructure.
Thank you. So our next speaker is David Wu. David is a MD PhD student at the Perelman School of Medicine at the University of Pennsylvania and is part of Penn's Genomics and Computational Biology graduate group. He studied computer science at Stanford before starting his MD PhD training. His research interests lie at the intersection between data science and medicine, where he hopes to apply computational techniques to large biomedical datasets to improve human health. David used STARR OMAP free text clinical notes to build a natural language processing tool that efficiently and accurately creates monogenic disease patient cohorts. David, please take it away.
Yeah, thank you so much for having me. I think I'm a little bit of an outlier because I'm not at Stanford right now, I'm actually calling from Philly. So it's nice to be talking to Stanford folks. I was at Stanford for undergrad, and that's the type of work I'll be talking about. So I'll be talking about discovering monogenic patients with a confirmed molecular diagnosis in millions of clinical notes. And this work was done under the supervision of Gil Bejarano, who's a professor at Stanford. So I'm trying to distill in this presentation one of the projects that I worked on while I was at Stanford. The full details of this particular work have been published. We published this in Genetics and Medicine late last year in October. So for any of those who are interested in diving into the details, I've provided the reference for this publication. I'll only have time to sort of briefly overview everything. So again, for those who are interested, the further details can be found in this publication. So I'll be talking about cohort building, which many of you are probably familiar with, but just for those who may not be super familiar with it, just want to define it as the process of collecting patients with some set of common attributes that you're interested in. So schematically, we all know that we have many patients' data in the Stanford healthcare system that we pull into an EHR, and we use STARR OMAP to access this information. And when I want to build a cohort, I'll probably come to the EHR system with some sort of query. A simple one is, give me all patients with disease X, because for whatever reason, interested in that population. And through the cohort building algorithms, hopefully the they can scan the EHR system and return a subset of all the patients that, in this case, have disease X. So there's a lot of opportunities that our lab thinks about when it comes to cohort building. I've listed a couple here. It can inform and improve the diagnosis and clinical care of patients, can aid in clinical trial recruitment, provide the basis for answering research questions, and in general, facilitate the implementation of evidence-based medicine. Our lab is particularly interested in monogenic diseases, that is, diseases that are caused by mutations in a single gene. You might have heard a couple of monogenic diseases like cystic fibrosis. And we think that cohort building and building tools that efficiently perform cohort building are especially important in the realm of monogenic diseases because these diseases are often very rare, which makes them extremely hard to diagnose and perform research on. So having a tool that can give you a lot of patients with these specific diseases would be really useful. Even though each of these diseases individually is very rare, in aggregate, they affect a good proportion of the population, about 5% of all human beings. So when we went to our clinical collaborators and asked, how would you currently, if I wanted a cohort of monogenic disease patients, how would I create this cohort in the EHR system? And I don't think this will be a surprise to anyone, but we got two sort of main ways. The first one is using ICD codes, which again is, I think, fairly standard. And the second one is because these diseases are caused by single gene mutations, you can simply search for that particular gene in the patient's clinical notes. So for example, if I want cystic fibrosis patients, then I can just search for CFTR, which is the causative gene for cystic fibrosis. And these two simple strategies are easy to implement and pretty straightforward. However, when we evaluated their performance on recovering monogenic disease patients, we found that much was left to be desired. First of all, ICD codes have very poor coverage of monogenic diseases. There's about 4,461 monogenic diseases that we know of, and that's represented by this red dotted line. And as you can see, I'm plotting the coverage of these diseases by the ICD codes. Very, very few monogenic diseases have some sort of ICD-9 or ICD-10 code associated with them. So that is really not a great sign for using ICD codes to recover these patients. Additionally, we ran some performance evaluations on precision, which is, out of all the patients that using ICD codes or searching the notes for causative genes gives me, how many of those patients actually have the disease that the ICD code says they do, or the gene search says they do. And we can see that, again, this probably isn't a surprise to most folks, but the precision is very low for ICD codes and causative gene symbol search. We also evaluated the recall. So we took 100 randomly manually curated patients from the EHR that we knew had a confirmed monogenic disease. And we asked the question, how many of these 100 patients can these two methodologies actually retrieve? And again, not surprisingly, ICD codes have poor recall because of their poor coverage of monogenic diseases. Additionally, causative gene symbol search, so again, searching the EHR for just gene mentions, has great recall, but again, at a very low precision. So these limitations of how we currently create monogenic disease cohorts motivated us to create a framework, which we call MonoMiner, to accurately and efficiently create monogenic disease patient cohorts from the clinical free text notes. So I'm going to go over the MonoMiner pipeline now. It's a little bit involved, so I'll try to explain things that are confusing. And also, I do want to get to the use case example that I have at the end, so I'll try to be a little bit faster in the review of how this pipeline works. So first, we take a patient and extract all the clinical free text notes, and then we split them into their component sentences, which is pretty standard for an NLP pipeline. And then we take each sentence and we try to identify whether it is useful to us. And so what does that mean? We try to identify two types of sentences that contain the clinical disease diagnosis of a patient, and sentences that also contain the genetic information. So what gene is mutated in this particular patient. And the way that we identify sentences that we think are useful to us is using a trigger word approach. So we have positive trigger words that we look for, and these words, such as year old, we've curated to indicate that the sentence does contain relevant disease or gene information. And we have negative trigger words that we look for that we don't like to see in sentences. For example, in the sentence number 3, father would be one of our negative trigger words. And those words we've curated to indicate to us that the sentence, if they're found in a sentence, the sentence does not contain relevant information. So for example, father, if a sentence has the word father in it, then it's probably not about the patient, it's probably about the patient's father. So we would not want to include that sentence in further analysis. So we search for sentences that contain clinical diagnoses and sentences that contain, again, the specific gene mutation mentions. The process is exactly the same for both of these sentence identification processes. And we look for sentences that have one positive trigger word and no negative trigger words. Specific trigger words that we use, sort of beyond the scope of this talk, but you can definitely read more about that in the paper. Okay, so now we've identified sentences that we think are potentially useful for us in diagnosing or extracting the diagnosis of this patient. Now what we do is we look for specific diagnosis information in those sentences. So for example, this is an example sentence, and it's fairly clear that the diagnosis is Marfan syndrome, which we identify using a dictionary of all monogenic disease names. And simultaneously, we look at the sentences again for specific gene mentions. So in this case, we found the gene FBN1, and so we collate that information together. So we found Marfan syndrome, we found FBN1, and the next thing to do is to ask whether this gene is consistent with the disease. So whether this gene causes the disease. And if it does, then we basically process this patient and send it to the last step of our monominor framework. And in this case, Marfan syndrome is known to be caused by FBN1 mutations. So this would be a success, and we would send that patient for further processing. So this is the last step of monominor. Once we have identified patients with genes and diseases that are consistent with a monogenic disease, the last step that we do is, for example, in this first patient, we'll extract the patient's phenotypes on the left, so all of the patient's symptoms, and we will also pull the known symptoms of the disease that we think they have. For example, in the first patient, cystic fibrosis. And there are algorithms to compare two sets of symptoms and see whether or not they are similar or consistent with each other. And so we use those off-the-shelf algorithms. And in the case for the first patient, it happens to be consistent with the diagnosis. So we formally diagnose this, or we formally annotate this patient to have cystic fibrosis. But as you can see in the second example, the patient's phenotypes or symptoms are not compatible with the disease that we think they have, Tay-Sachs disease. And so we would reject this potential assignment. And so that's how the monominor framework works. And so, I've explained this complex pipeline. How does it actually do on performance? So I've added the performance of our pipeline compared to ICD-10 codes and gene search that I showed you previously. And we were really happy to see that our monominor pipeline has greatly improved precision. So the patients that it pulls out actually do have the disease that we think they do. And achieves a moderate recall that's between ICD-10 code search and gene symbol search. But we were really happy about the precision. And that was the main criteria we wanted to optimize. So we were okay with paying a little bit of cost to the recall. So now I think this is the most important slide, is an example of why these cohorts that we create are useful. So here's an example of a cohort that we pulled from the Stanford medical record database for hemochromatosis patients. So this is a disease where your iron levels are elevated and that causes a lot of problems. And so what I've plotted for each patient, which is represented in a graph, is a time course of their iron level. So iron level is on the y-axis and time is on the x-axis. And I've normalized the time to 0, representing the time of the first recorded treatment of these patients for their hemochromatosis. And what you can generally appreciate, I hope, is that the iron levels start out very elevated, for example, in this patient 2, before they start treatment. But once we start treatment for their hemochromatosis, the iron levels quickly fall and to a more acceptable level. So that's what we want to see. This is encouraging. So after we treat these patients, they should get better, which they do. However, what you'll notice is some patients have a trajectory that is somewhat concerning. For example, this patient number 4 has a drop in iron as they are started to get treated. But as you can see, it's the iron sort of creeps up again to a level that it was before the treatment, which is concerning. And so digging into this patient further, which is, I've blown up the patient's graph on the bottom of the slide. We've actually saw that the patient, they started treatment with the red dashed line at time 0 and their iron fell. But at some point, for whatever reason, we found out that they stopped getting treated for their disease. And a number of years after they got stopped on the treatment, you can see they came back to the healthcare system with a markedly elevated iron levels. And so were restarted and those iron levels fell back down. So I think this is a good example of how you can use maybe these cohorts that we're creating to basically do quality improvement. Ask questions like, why was patient 4 stopped on their treatment? And why did they not follow up so often? As you can see between the blue line and the green line, they only got 1 measurement of iron. So what happened in that time? Is there anything that we could do in the healthcare system to not have sort of these situations where patients are lost for a long period of time and then come back with the disease as bad as it was when they first started. So in conclusion, I hope I've convinced you that monominor allows the creation of high quality monogenic disease cohorts that have clinical and research utility. I also want to point out that monominor is highly portable due to its use of clinical free text notes only. So it doesn't use anything else, just uses clinical notes. And clinical notes are basically the bread and butter of how doctors document information about their patients. And lastly, monominor's framework, although our lab was specifically interested in genetic disease, specifically monogenic diseases, this framework we think can be potentially adjusted to capture other disease classes as well. So with that, I'll conclude my talk and really thank everyone involved in this work, specifically Gil Bejarano, who was my PI when I was an undergrad at Stanford in the lab, John Burstein in the Division of Medical Genetics, our clinical and who are our clinical collaborators who are really helpful in providing clinical advice. And as always, the Stanford Research Computing Center with NIGM and others for providing such a rich data set in the STARR OMOP resource for us to develop these tools. So with that, I'll thank everyone for their time and happy to answer any questions.
David, is the code for this open source?
Yeah, so this code is open source. I don't have the GitHub repository link here, but it should be in the paper if you're interested. And yeah, everything is open source, so you can take a look if that is of interest.
Thank you, David. There is one question from Devanish. So his question is, how many monogenic diseases does it mine currently?
Yeah, so that's a good question. We extracted all of the diseases in the OMIN resource. So it's sort of the comprehensive resource for genetic diseases, and we curated the OMIM. There's many thousands of diseases in OMIM, and some of them are monogenic and some of them are not. And I didn't go into this specifically in the presentation, but it's in the paper if you're interested. But we spent a lot of time thinking about how to classify diseases in OMIM as monogenic or not monogenic, and we extract approximately, again, I think I mentioned it in the presentation, about 4,500-ish monogenic diseases. So monominer is able to capture any of those 4,500 diseases. See, there's another question from Monica. How many models for significant words for symptom, gene, mutation, sentences were trained? The ones used in the initial parts. Yeah, so I didn't get into this either super specifically, but in order to figure out the trigger words that we used and the general framework of monominer, we trained it on, I think it was, we hand curated, for the recall set, we hand curated 100 patients. So we read, we just read a bunch of medical records and found 100 patients with confirmed monogenic diseases. And for the precision set, we ran, the algorithm on, I think, a subset of Stanford hospital system patients. I think it was either 100,000 or 200,000. I forget now the exact details. And then we sort of optimized the parameters on that training set, and then applied it to the whole EHR system as evaluation. So Oshra asks, can you please say a little bit about how this project is planned to continue at the lab? This is a good question that I frankly don't have an answer to. So I left for Penn quite soon after sort of I wrapped up this paper. And so I don't know specifically what Gil and the team has planned for this project in the future, but I could definitely sort of reach out and ask a little bit about that. Any other questions?
David, this is Anup. I have a question. First of all, fantastic work. The question is on slide 15, if you were to go back, you noted that
Right.
in patient 1, for example, you're looking at patient phenotypes that you're expecting for the cystic fibrosis patient. And then you're looking at a record and then basically looking at those symptoms and saying, well, it's consistent versus non-consistent in patient 2, for example. Now, what if the patient has not all the symptoms, but just a few?
Right.
Would that then classify, like how would the classification work then?
Right. So there are sort of, I think I briefly alluded to this, but there are sort of off-the-shelf tools actually developed in Gil's lab that sort of take as input 2 sets of symptoms, and they will output some sort of score on how similar, the score, I don't know the exact details, but it might be like 1 to 1,000 or 1 to 100. The higher the score, the more similar. So, partial overlap does have a score, a decently high score, maybe not 100, maybe not, totally matching, but it might be like 50 or 75 or 25, depending on how many symptoms overlap. And it also compares the type of symptom. So if I have a respiratory symptom, such as cough, and then, for example, cystic fibrosis is known to cause another respiratory symptom, bronchitis, then those 2 terms, even though they're not the same exact symptom, there is some similarity around them, and the algorithms take into account. And so there's certain score cutoffs that you can set to have a notion of similar enough, if that makes sense. Because we don't want, just patients that have the very cookie cutter definition of cystic fibrosis, because we do want to be able to expand our cohorts to those that maybe have atypical symptoms or only have some of the symptoms and have other symptoms, because we know that patients with these diseases are not, homogeneous. So there is some cutoffs that you can optimize to define just how similar is similar enough. Does that help?
Absolutely. Thank you. Thank you very much for that explanation. My second question is, does monominer allow you to use, for example, regular expressions to be able to query the existing free text nodes?
Yeah, so we like our trigger word sort of framework sort of is like a regular expression. It's not exactly a regular expression, but I imagine if you wanted to change some stuff in the specific wordings that you want to accept, or you want to add new trigger words or remove trigger words, you can certainly do that. But whether to answer your specific question, like, can you use regular expressions? Can you like give monominer regular expressions to look for? That's not a feature that we have currently.
Okay, got it. Sounds good. Thank you very much.
Yeah.
Fantastic work once again.
And thank you so much. And then the last question I think I see in the chat is, did you create these symptom frameworks for all of those 4,000 diseases? And the answer to that is we did not actually have to create the frameworks, because there is a database for the human phenotype ontology, which is the symptom ontology that we use. And they have already annotated all of the monogenic disease, or many of the monogenic diseases, with a list of known symptoms for those. So that's available publicly, and so we would, that would be a lot of work for sure to manually curate the symptoms for 4,000 diseases. But luckily, that was available to us as a public resource.
Thank you so much for all those great questions. And Dave, thank you for your wonderful presentation.
Thank you so much. Thanks for having me.
Yeah, you're welcome. We would like to move on to the next speaker, Dr. Alex Sandhu. Alex is a cardiologist with interest in the care of patients with advanced heart failure. In the year 2022, Dr. Sandhu collaborated with research technology team members, Alvaro Perez, Lian Yasukawa, myself, and few others, in the use of real-time alerting integration with REDCap to identify predictors of pro-HF trial enrollment among eligible patients seen in the Stanford Heart Failure Clinic. Alex, take it away, please.
Thank you, Sanjay, for the introduction. So my talk will be a little bit different in terms of kind of how STARR and Research IT helped me with my project. As very brief clinical background, when we talk to patients in the clinic to assess how they're doing with any condition, including heart failure, which is my condition of interest, we predominantly base it on a series of unstructured questions, an interview kind of asking what their symptoms are and how they feel. And there's kind of a vast and expanding amount of data that, rather than have the clinician interpret it, if you have the patients complete an objective questionnaire, patient-reported outcome measure, you can capture data that's better prognostic and more reliable than actually feeding it through the clinician interpretation. So based off of that, there have been kind of cardiology society guidelines that I was a part of that recommended that we start collecting patient-reported health status to help guide clinical care and understand prognosis. But there's no randomized data to help us understand how collecting that data improves or even impacts clinical care. So that's what we set out to do in the PRO-HF trial. We basically wanted to understand how collecting patient-reported health status using a validated questionnaire, in this case the Kansas City Cardiomyopathy Questionnaire, impacts the care of patients in the heart failure clinic. And I won't go into too much detail, but our primary outcome was kind of the impact on patient-reported health status 1 year later, and then also evaluated a series of secondary outcomes, including how accurate the clinician assessment of health status was, how well it aligned with patient-reported health status, and the patient's experience in clinic. So this was funded as a K-23, and to give you a little bit of background on why we had to think of kind of a very innovative enrollment strategy, it was funded as a K-23 Career Development Award from the NIH, which is a generous award in terms of protecting my time, but gives very little in the way of money for a clinical trial. less than $50,000. And initially, it was done with the expectation after some kind of brief discussions that this would use waiver of consent. And so with waiver of consent, we plan to basically deliver this survey to 50% of all patients being seen in the heart failure clinic, and then kind of compare how things kind of ended up for the patients that got the questionnaire versus those that didn't. And for reasons that I think are a little bit surprising, and honestly probably a little bit of a misinterpretation of the common rule, the IRB ended up denying our waiver of consent and actually suggesting that rather than do this as research, why don't we just do it as quality improvement? Then we won't need to necessarily use consent, and it really does meet the standards of quality improvement. So then we went back to NIH and we said, okay, well, can we do this as quality improvement? And they said, well, if you want us to give back, if you want to give back our money, then you can do it as quality improvement. So in that case, we were kind of stuck having to do the clinical trial. So we then asked NIH, can we reduce the sample size? And they also said, no, that's kind of what you told us we we're going to do. So we basically had to find a way to recruit and consent 1,200 plus patients, which was our initial sample size, with very limited funds. And that was kind of what led to this, I think, very fruitful collaboration with Research IT, and I'll walk you through how we did that recruitment process. So what we needed to do is we needed to identify eligible patients being seen in the heart failure clinic near real time when they kind of scheduled their visits. We needed to be able to identify them and then subsequently contact them and consent them virtually, hopefully without even necessarily needing any sort of human contact, given this is really a minimal risk, simple intervention. And then after we consented them, we needed to randomize those individuals into one of two arms, either an arm where they get the patient-reported health status assessment or one where with usual care. And for patients in usual care, we actually needed to deliver the assessment real time, because we knew that if we didn't deliver it real time, if we later reached out and delivered it, there would be a very low completion rate. And then after enrollment, we needed to kind of track patient data. So I'll walk you through kind of how this was done with the help of Leanne, Alvaro, Sanjay, Andrew Martin, and others. So first, what we did is they built us a link between Epic Clarity and REDCap to actually real-time import any patient appointments that were made in the Stanford Heart Failure Clinic. So we could see when individuals were making appointments, and then based on that, kind of determine what the outreach would be. That not only pulled in the appointment, but it also pulled in the providers to make sure that they were eligible providers. And then from STARR, we pulled in additional kind of clinical data to do some amount of light filtering for which patients were eligible based on their clinical characteristics. So from that, we were able to kind of automate the recruitment and enrollment process. So we set up automated emails, and patients were emailed about 8 to 11 days before their visit, asking them if they're interested in enrollment, and from that they'd either be able to decline or they'd be able to click yes, do an electronic consent, and then right after that have the intervention delivered. For patients that didn't respond to the email, either affirmative or negative, we actually had text messages sent, also automated from REDCap, to try to enroll them via text message or to get if they were interested, and then our study coordinators could actually follow up with them afterwards via call, but with not a cold call, with a warm call where they're expecting it after the text message. And then, as mentioned, for individuals that neither declined nor enrolled via either the email or text message options, we then had our study coordinators reach out via phone, which we thought was important not only to kind of augment enrollment, but also to capture those patients that might be less likely to enroll via email alone. And, telephone enrollment alone for the 1,200 patients with the budget we had wouldn't have been feasible. But as I think I'll show you later, we were basically able to enroll 700 patients without any communication from the research coordinators that not only enrolled, but then completed the initial surveys that we asked them to. And so our research coordinators were then able to focus on enrolling another 600 individuals and we were able to enroll 1,300 patients in our trial over just a 9-month period. Post-enrollment, as I mentioned, it was really important to kind of deliver the intervention incredibly efficiently. So we had kind of automated real-time randomization. So immediately after they clicked consent, there was block randomization stratified by the treating clinician, which we knew because we were importing that data from Clarity for who they were going to see in the clinic. They were assigned to either the usual care arm or the KCT12 arm. In the usual care arm, we actually were not making these patient-reported health status assessments available to the clinician, but we still wanted to collect a baseline to understand change in this over time. And so we actually, right after they consented, it would go to a screen where, on the web, they would complete the KCT12. And that was a huge help because otherwise it would have been a ton of work for our research coordinators to get a hold of them again to get them to complete that. For patients that were randomized to the KCT12 arm, we actually completed the instrument in Epic. And so there, our research coordinators basically had a filter list for everybody daily that was randomized to the KCT12 arm. They would enter a dot phrase in Epic that would then assign that patient to get the KCQ before every one of their heart failure clinic visits. So that's, not necessarily what we're talking about today, but a whole different aspect from the research IT team of how they enabled this trial to be possible. And then after the patients were seen in their first clinic visit, we also used GuideCap to automate transmission of post-visit experience surveys via both text message and email to understand how the patients felt about their clinic visit, and ended up having response rates of about, I think, 75 to 80%, so quite high. And then finally, much of our follow-up data is actually just being collected via a STARR to REDCap link. So we're collecting all the data about medication changes, encounter frequency, and subsequent KCQs via that STARR to REDCap link. This is just a quick schematic for some of the other work that the IT team did. I guess this is the research IT team so much as Joker's team, where they not only built the KCQ into Epic, but they built mechanisms so that the clinician can actually readily visualize this information in Epic to make it really impactful, so it actually could influence care to basically stimulate the clinicians to use this information to develop dot phrases and visualizations. And then we supplemented that by printing out the results of that patient-reported health status instrument. This is the kind of overarching study design. We had 5,000 patients seen in the Stanford Heart Failure Clinic. I think after including 370 individuals who were in kind of alternate trials. And then after this enrollment process, which was both the email, text, and telephone, we enrolled 1,249 individuals. And as I mentioned, more than half of those were enrolled from the email alone without requiring a text message or a phone call. And then subsequently, we collected the outcomes that I'll go over. I thought this was quite interesting. This is a paper that is under review at a journal right now, where we tried to understand how representative the sample was that we enrolled in this kind of virtual enrollment. And we did see disparities between who we enrolled in the trial and who we didn't. But I think the magnitude of disparities we saw were actually much smaller than what you see from most traditional clinical trials where all enrollment is done in person. Overall, the consented population had a relatively similar age compared to the non-consented population. We saw that female sex, which is traditionally far underrepresented in cardiovascular trials, was similar to the proportion in the overall clinic population. We did see racial and ethnic differences, but again, I think probably smaller than you see in most traditional clinical trials. One of the things I didn't share here that we also found as we looked at which proportion of patients were enrolled via email versus were enrolled with a phone call visit. And I think what we saw was, the severity of heart failure was pretty similar across both. Actually, the population that was enrolled via phone call the population that was enrolled with email alone versus email plus phone call, the population that was enrolled by email alone was actually older than the population that was enrolled with email and phone call, which I think was somewhat surprising. And then the Hispanic population had a far lower enrollment rate with email alone compared with email and phone call. And some of that actually speaks to some proportion of this Hispanic population having a lower email rate in the system. So I have some preliminary results, and I can say that they were quite positive for the intervention. I think that's of less interest for this. I think I can stop here, and just huge thanks to the team of Leanne, Sanjay, Alvaro, Andrew, Todd, and everybody else. I mean, I think the results of the preliminary results of this trial were shown as a late-breaking clinical trial on the big stage at the American Heart Association. We already have 3 publications from this trial and another 2 that are in press. The final results are still pending. But I think what over and over is kind of a takeaway from colleagues around the country when I talk about this is they're amazed at the operational ability to pull this off, both in terms of the trial and in terms of our ability to collect patient-reported outcomes at scale. And I think that just speaks volumes to your guys' team and all the support you're able to give for this.
Thank you so much, Alex, for that presentation. I have a quick question. Would you be able to talk to us briefly about how the use of real-time alerting plus REDCap reduced your recruitment costs?
Yeah. I don't have an exact estimate, but like the traditional clinical trial approach just wouldn't have worked for this trial because we needed to contact people before their first visit. I think the cold approach of having coordinators do all of the calling probably would have required us having 2 to 3 times the amount of coordinator support that we had. So I think that would have probably doubled or tripled the cost of the trial easily.
Wow, that's impressive. Thank you so much, Alex, for the wonderful presentation. Let's move on to, if there are no further questions, we can move on to the next presentation. Our next presenter is none other than Dr. Anup Rao. Anup's specialty is neonatal perinatal medicine. His research interests include innovation in health tech, wearable sensors at affordable costs. Over last years, among other things, we've collaborated on bedside monitoring data from NICU in testing the efficacy of a non-invasive sensor. Anup, take it away, please.
Yeah, thank you very much. Again, thank you very much for this opportunity. I'm really happy to be here to share some results of work that would not have been possible if not for the tremendous amount of help and support that I got from STARR and the various incarnations of STARR. The work was also supported by an NIH R43 grant and also support from FDA and internally from the Maternal Child Health Research Institute. So my disclosures for today, I'll be talking about an investigational device that has not yet been approved by the FDA. So I am very privileged to work with very small babies and babies who really don't stand a chance to begin with, but because of the incredible team, because of the incredible resources that we have at our disposal, and also some excellent colleagues who are going to follow me, Anne and Jeff, we're able to actually provide a lot for these babies. What also helps us is we have incredible resources in terms of a ton of sensors that are at our disposal. But more often than not, what happens is all these data are really lost, and we usually look at these data several years down the line in terms of retrospective studies. So I think I consider that as a crime. And the question is, can we use all of these data in real time? But before I move ahead, let me kind of set the stage here and say, well, for the next couple of minutes, I'm going to talk about two examples, work that we have already completed and some work that we are in the process of doing. And in both cases, STARR has helped us quite a bit. So the first example is that of a non-invasive continuous blood pressure sensor. Why did we ever start working on this thing? So one of the challenges we have is when we have very small babies, we have to monitor their blood pressure. We insert a catheter into the baby's wrist or foot or the belly button. Now, just to give you a sense of proportion, some of our babies are very, very small. Their wrists are about the size of your thumb. And imagine trying to put a catheter inside that. Now, if you can get the catheter in, great, we can monitor the baby. If you do not get the catheter in, that's a challenge, but we'll try some alternative ways to get arterial lines into other places in the body. But here's something to think about. This is a baby. We got this baby referred to us at some point, maybe about 7 years ago. This baby got referred from a different hospital, and somebody had stuck a catheter in the baby's belly button, as is usually the case. And turns out the leg had turned black. No chance of recovery. There was no pulse whatsoever. Essentially, the baby ended up having a below-knee amputation. So these arterial lines have benefits, but they have problems as well. So the question was, hey, can we do something so that we can turn this invasive procedure into a non-invasive procedure? And can we continuously and non-invasively monitor blood pressure in neonates? So with that question in mind, we ran into two wonderful individuals on campus. This is a great example of a cross-campus collaboration. Professor Janan Bao and Dr. Zina Kwan, both chemical engineers. And Dr. Zina Kwan went on to start a company called Pyramis. So what Dr. Janan Bao's lab has expertise in is sensors. And she'd come up with this sensor with, a tiny sensor that was stuck to a Band-Aid that would be able to pick up pulsations. And Dr. Zina Kwan then took this technology and then built a company out of it. And this is one of the first prototypes of the sensor that was adapted for neonates. It's completely wireless. And it's just, think of it just like a wristwatch. That shiny rectangular portion in there is the sensor. And it's a capacitance sensor. And a good example of capacitance sensing is, your modern day phones, which even with the slightest of touch, the screen lights up. And in our case, what happens is the slightest of variation of the pulse on the surface of the skin gets picked up as a capacitance signal that varies over time. So this is what happens when the sensor is wrapped around the wrist. The tiny little pulsations that you see here, hopefully, are picked up by the capacitance sensor. And then all of this is processed. And I hope you can see the screen here. Let me minimize a few other obstructing things here. So these data are processed. And why is processing important? Because this is an extremely sensitive sensor, machines that the baby is attached to can interfere and corrupt the signal. And so you need to filter these things out. Now, in addition to filtering, it's very important to note that the signal that you're seeing is a very unscaled signal. So it's an unscaled waveform. And for us to be able to convert an unscaled waveform into a waveform representative of the blood pressure, it needs to undergo further processing. And here's where STARR's effort helped significantly. We were able to create a database of more than 300 patients with more than about 25,000 hours of data, and sort of used artificial neural networks to be able to train several blood pressure estimation models, which allowed us to take an unscaled waveform, look at that, and convert that into a scaled waveform representative of the blood pressure. So I think this is where I interacted with the STARR's team, and we were able to, over a fairly long period of time, figure out how to get this process going. Long story short, we already have the product. This is sort of version 3, version 4, you can call it whatever you want, but this is sort of the final product that we have. Sensor wrapped around the wrist or foot of a baby and wirelessly transmitting data, computing on the fly to be able to get the blood pressure for the baby in real time, available at the bedside. This device, among other things, got the FDA breakthrough device designation, and allowed us to make tons of improvements. We've done several clinical studies, not just in neonates, but also in adults, because this is platform technology, all built on sort of the intelligence that is there behind the artificial neural network model that allows us to estimate the blood pressure. All the technology aside, I think, again, it gives us immense pleasure when some of these things are actually used in practice. And so this is one of the babies that was born in 2019. I encourage you to appreciate the proportion of the baby vis-a-vis everything else that you see in the photograph, to be able to understand that some of the data-driven efforts, the technicalities behind how you get these models to work, and the wonderful discussions that all speakers before me and after me will talk about, ultimately, these things are meant to help patients. And this is a great example of that. That baby there was one of the participants in our study. And so you can see how it makes us do things well on a daily basis. This is example number 1. This device and our multi-center data for neonatal work has already been submitted to the FDA, and we are waiting for some kind of positive decision from them in the coming months. Let me move on to another example. This is a mouthful, but so what we're looking at is heart rate variability in a condition seen in babies called as hypoxic ischemic encephalopathy. It's a $5 term for saying babies around the time of birth may have less oxygen delivered to their organs, or less blood flow, and hence less oxygen delivered to their organs as a result of some event that is multifactorial, but the end result is the following. Babies' brains get affected quite a bit with even the slightest disruption in oxygen delivery. So we worry about it, and that's why we call it encephalopathy. But so are other organs. Other organs get affected too. The heart gets affected, the liver gets affected, the kidney gets affected. But the challenge is, once these babies come out, they can have varying levels of activity while still having brain damage. So when the parent stares at you and asks you, how is my baby going to do? You can only shrug your shoulder, because any outcome data is going to be available later on in life. For example, we do an MRI at around 5 to 7 days of life. But until then, we'll never be able to tell a parent how the baby is going to do. So what's the standard of care for babies who are born with HIE? The standard of care is something called as therapeutic hypothermia, where you cool the baby. You cool the baby down by a few degrees for about 3 days. And I can show this to you pictorially. You cool the baby down for 3 days, and then you rewarm the baby at the end of 3-day period. And then essentially hope for the best, try to fix whatever problems you're encountering. And then around 5 to 7 days of life, you end up getting an MRI to understand how much the brain got affected. If someone's lucky, the brain escapes unscathed. And if someone's unlucky, the other extreme is devastating injury, or the baby's organ systems have failed to that extent. Every organ system would have failed to an extent that the baby dies. So the question we asked here is, are there ways in which we can utilize some of the heart rate variability features during this period, which is the cooling period, and be able to predict the outcome at around 1 week of life. This is work in progress. So I just preface it that way. But this was the question we were asking. We were also asking, is there any blood pressure variability that is associated with eventual outcome as well? And we also compute something called as near-infrared spectroscopy. We have that data as well. But this is sort of the overarching pipeline for how we collect and store data before we process it. The ECG and arterial blood pressure waveform data through the Stanford warehouse and the STARR services that are offered. And the near-infrared spectroscopy data goes to a device, doesn't go into the data warehouse. You got to manually download the data. So we created a waveform database. And at least in our preliminary data set, we looked at about 140 patients or so. And this is sort of the overall pipeline for just the ECG and blood pressure processing. The near-infrared spectroscopy processing is a little bit easier. Overall, for the ECG, we started off with the beat-to-beat intervals. These are the numerics files that we obtained from the STARR group. Using some Python scripts, we were able to calculate the time, frequency, and nonlinear characteristics. We then ran it through a regression tree model to be able to achieve outcome, which is like, life or death survival outcome. And our accuracy was around 92%. Again, preliminary data still need a little bit of digging into to understand where some of the sources of error are and how this can be improved and what the potential sources of confounding might be. Similar analysis for blood pressure. This is the raw data that we obtain, and we're able to calculate the blood pressure variability metrics. I have not looked at the blood pressure variability data for these patients, but that would essentially be my next step. So I think overall, I wanted to share two examples. One, where we have already completed the project, and the project is like far out there. We've published now about 2 papers, have had multiple presentations at conferences. As I said, we successfully completed the NIH R43 grant here, and we were able to get a tremendous amount of support in terms of being able to utilize these devices in different settings. It also supported a lot of academia industry cooperation. And the second example where we are able to predict mortality versus survival for a specific subset of neonates who have this terrible condition called HIE. So overall, I must thank STARR for helping us solve some, not so small problems. A large team that has helped over the last 6 or 7 years, and tons of supporters both inside and outside the institution. And my specific thanks to Sanjay, Lee, Priya, Somali, and Stephanie for helping us do what we aspire to do. Thank you.
Thank you so much, Anup, for such a powerful presentation. I honestly cannot imagine more impactful work. So thank you so much. Unless there are questions.
That was really cool. I had a really quick question, which is, why do you cool the baby? Is that because of the inflammation or?
So cooling the baby allows us to slow down the metabolic processes. And slowing down the metabolic processes allows the body to heal to some extent. And as you would expect, some babies heal better than others for unknown reasons. And this treatment has proven, or at least there is data to suggest that the overall neurodevelopmental outcomes are better when cooled as opposed to not cooling. And just to put this in context again, when a baby is being cooled, some of the babies don't move at all, and they can turn out to be okay or not okay. But when parents are having a conversation with us about how is my baby going to do, as I said, currently we shrug our shoulders, and with your help and with the help of the data, as you can clearly see, in the first 3 days of life, with about 90+% confidence, I can tell you overall which direction the baby is going in. I don't know if it's a good thing or a bad thing. This is where some of the ethical questions that were raised earlier come up. And also one very important point, I mean, the fact that you have the data doesn't mean you have to do something with it. But this allows us to get us to think more mechanistically about what is it that is in the heart rate features alone, no other data. I don't know if it's a male or a female. I don't know how old the baby is. I don't know ethnicity of the baby. I don't know any biochemical parameters of the baby. I don't know the MRI of the baby. But just with the heart rate variability features alone, agnostic of any other pieces of information, you're able to make that prediction of, is this baby going to live or die? And that has tremendous implications, especially if you go outside the US, where you don't have the fancy equipment, you don't have the fancy, 10 times a day, let's measure this variable and try to see what is happening just with ECG alone. So I think it is powerful, but I kind of digress, but just wanted to share with you how STARR has helped move the needle quite a bit.
I think there's a question in the Q&A.
So there is one question from Dr. Steffner. So she's asking, were you using the raw waveforms from the bedside monitors or just the numerical values? I think by raw waveforms, she's talking about the .dat files. And the second part of the question is, were you limited in any way by having to use a proprietary viewer, or did you use open source viewer to just spot check the data?
Yeah. So Kirsten, thank you very much. So your first question was, yes, I'm using the numerics data for the beat to beat RR interval, but for the blood pressure data, I'm using the .dat file, which is the raw file. As far as viewing, I think there are many options, but I usually use Python, so allows me to just plot and take a look at the data to see what is happening. So am I limited by the proprietary viewers? No. But then proprietary viewers have their it'll cut short your time to process the data. But I think having being able to do it on your own allows you to have that independence to play around with the data like nothing else. Be able to visualize, be able to turn it around, transform it, use different ways of being able to process it, try to time sync it, all issues that one ends up facing. Having said that, I think the proprietary viewer is great to just kind of take a look to see if everything looks okay. Hopefully that answered your question, Weston.
Thank you.
Quick question, the PhysioNet, like when you convert the files into PhysioNet, is there not an open source viewer to view those files, or?
I am sure, Priya, there is. I just haven't played around with it as much.
Okay.
And I could be at fault for it, but I just wanted to make sure, because most of the time what happens is our challenge is, once you're able to view the data, then you still have to play around with it. I think the money is in playing around with it part. So I must say, my ignorance shining through. Is there an open source viewer? I am 100% sure there is. Have I used it? Absolutely not. But I'd be happy to look around for you and just send you an email with what may or may not work. Or happy to share, whatever code I have so that you can play around with it.
Thank you.
Mm-hmm.
Yeah, thank you, Anup. That was going to be my question. I was trying to read Dr. Steffner's mind. That would you be willing to donate the code to her lab so she can use your view Python?
Absolutely. The biggest disclaimer being I'm pretty much a coder at gunpoint. So if you look at the code, don't laugh at it. And please feel free, and a huge disclaimer there. So I'm more than happy to share so that you can, play around with it, do whatever you want with it. Anything for kids or adults. I have nothing against adults, but
Thank you. Thank you so much, Anu.
no problem.
With that, let's move on to the next team of speakers. So Dr. Jeffrey Yang and Dr. Ann Taylor are going to be doing a team presentation. So Dr. Yang is a fellow in pediatric cardiology and will be doing an additional fellowship in pediatric ICU medicine. Dr. Ann Taylor is a fellow in pediatric interventional cardiology and will be faculty at Children's Colorado. We collaborated on telemetry review following transcatheter pulmonary valve replacement. Dr. Yang also performed heart rate variability and alarm fatigue studies. Take it away, please.
Thank you.
Hi, I'm Jeff. This is Ann. We've worked with STARR a few times. We worked with Sanjay a few times using kind of waveform data as well as kind of the STARR wave data lake to kind of help us with various QI and other patient safety and kind of tools for improving our clinical effectiveness. So we've kind of just put together three examples of how we used it. I think we'll focus probably more on the first two, and if we get to the third one, if time allows. Here's just the two of us. Thank you for your interest, Sanjay. And so, like we talked about, bedside monitoring telemetry has just a ton of clinical information. I think Anu brought it up earlier, so did many of the other speakers here. And I think that data can, obviously helps us take care of our patients, but also we can be inundated with data. And so a lot of what research does is try to figure out how can we use that data to improve patient safety and how we take care of them. So here are three studies. The first study we'll talk about is the one Ann and I looked at for ventricular arrhythmias after a transcatheter pulmonary valve placement or harmony valve placement. And then we have a study on alarm fatigue as well as heart rate variability in the pediatric ICU setting. So I think Ann's going to start off talking about the first study.
Yep. So I'll give you guys a little bit of background because I know we have a mix of clinical folks and non-clinical folks on the Zoom. So this project was aimed at looking at the burden of ventricular arrhythmia following pulmonary valve replacement in the cath lab, so transcatheter pulmonary valve. So this is predominantly done in patients who are born with tetralogy of Fallot, in which the pulmonary valve is small and anteriorly deviated. So this is just a schematic of that. And then quickly, I'll show you guys what the common surgery was and still is for some of these patients, is to kind of cut along that narrowed valve and then open it up with a patch. We call this a transannular patch. And that repairs the patient for some time, but leaves them with kind of to and fro flow across their pulmonary valve, which over time dilates their right ventricle and leads to the need for a new valve to be placed. This is just an echocardiogram showing the probe is on the patient's chest. We're looking at the pulmonary valve, which is going from the top of the screen, and then the flow is going across the valve, which is right here, if you can see my cursor, and then down to the pulmonary arteries. And what this image is demonstrating for you guys is that this valve is leaking. So there's the blue flow, which is normal, going away from the chest, going down from the right ventricle to the pulmonary arteries. And then there's the red flow going back into the ventricle, which is abnormal. And so this is a patient who over time, their right ventricle, which is the right pumping chamber of the heart, dilates, and there's a need to replace this pulmonary valve. And I'll give you guys a little bit of background on that. Here's just a picture of that right ventricle. It's this chamber on the left side of the picture, and that's rather large. Over time, these patients develop right heart failure, and they can develop arrhythmias. They can ultimately have exercise restriction, and then they're at risk of sudden death as well. So this is a big deal in a large population of patients who were repaired at birth. This is an MRI, just again demonstrating that big right ventricle. So a little bit of history about transcatheter pulmonary valves. In 2000, the first ever transcatheter pulmonary valve was devised, and it's made out of bovine internal jugular veins, because those veins actually have a valve in them. So you can take the bovine vein, use that valve, implant it in the position that the patient needs, which in this case is the pulmonary position. And then suddenly you have a working valve that doesn't allow the blood to leak backwards. So this was done in France in 2000, and this was the first pulmonary valve. And then since then we've come a long ways. We have on the left side, we have the Edwards Sapien valve. And then on the right side, we have the Medtronic Melody valve. And then going forward, for large dilated pulmonary valves here in those patients I was talking about before that have what's called a transannular patch when they're a baby, we have new valves that are called the Harmony valve, which is Medtronic, and then there's the Altera valve, which is Edwards. And for the purposes of this talk, the details of this don't particularly matter, but the point is to say that when we place these valves, the patients are at risk of having ventricular arrhythmia. And they found in their study, in the early clinical feasibility trial of these valves, that some patients, 14% of patients, did have ventricular arrhythmia, meaning an electrical impulse coming from the ventricle that generates a fast heart rhythm that can be dangerous and even deadly. So it's something we have to pay close attention to for these patients. We had a 40-year-old patient back in September of 2021 who is exactly as I described. He's the type of patient that when he was a baby, he had the transannular patch placed. It led to a dilated, leaky valve. His right ventricle dilated. He had exercise intolerance. He needed repair of the valve. We placed what's called a Harmony valve in this location. And then following the valve placement, he actually had a cardiac arrest on the acute care cardiology floor. So after this, the patient luckily survived, but had significant morbidity. He had a liver laceration during his CPR. He required mechanical circulatory support, which is like a bypass machine to keep his heart and lungs working while he recovered. He was in the hospital for months, and he is subsequently better. But this event caused us to say, we need to be more knowledgeable about what patients are at risk and how much ventricular arrhythmia do they have after these valves are placed. We want to know why this happened. Are all of our patients that get this at risk? And what data do we really have to answer this question? And that's where the telemetry database comes in for us. So back in fall of 2021, Jeff and I reviewed all of the telemetry data for 30 patients that had undergone this procedure. And so that's where your guys' group comes in. We were able to get access with all of your help to the Philips telemetry database, and we were able to go one patient at a time and pull their entire telemetry history from the time the patient was transferred from the recovery suite in the PACU to our floor or to our ICU. We were able to look at any single heartbeat that the patient had during their stay, we could review. So Jeff and I kind of painstakingly went through every single patient's entire telemetry duration for their stay. Luckily for us, they typically stay about 2 days and go home. But this meant Jeff and I really clicked through every single heartbeat for every single patient. So it was quite a bit of work. We're grateful that we had access to this data. I think, you know, as AI progresses and people are able to write algorithms to kind of identify this without a human going through it, I think we'll be able to do even more with telemetry data. But right now that doesn't exist reliably, so we had to go through it by hand. And so what you see here is these beats at the top are all of the heartbeats, and you see lots of normal heartbeats, which are these narrow looking kind of skinny vertical lines. Those are normal. And then as you get to the end, you start to see some abnormal looking beats. These wide beats are abnormal beats coming from the ventricle. And then as we progress through this patient's study, suddenly we start to see a couple of wide beats in a row. And so that is the beginning of a ventricular tachycardia. And when there's 4 beats or more, we call it a non-sustained ventricular tachycardia. And so what we did is we actually documented every single one of these episodes that the patients had. And then we went through and kind of cataloged them and said which ones were the longest and the fastest, and kind of the most concerning looking. And then we were able to compare that data that we collected from the telemetry to go back and compare it to the patient's demographics, their clinical history, their prior arrhythmia, their arrhythmia when they went home and had patch monitors on. So we were able to look at everything and run the statistics on that. And we actually were able to publish all of that data about a year ago. In addition to going through all of the tele, the Philips database gives us the classification of every single heartbeat. So we were able to look at these files that Philips spits out and say every single beat is labeled. And if it's labeled as a ventricular beat that's not supposed to originate in the ventricle, then this label, instead of saying I, would say V. And so we were able to calculate the number of abnormal ventricular impulses that the patient had. And so that kind of further contextualized our data. This is just an example of some of the rhythm strips that we found from the Philips database. And this is the figure that ended up in our paper. The top one is the patient who had a cardiac arrest. You can see that this is a very abnormal looking wide complex tachycardia is what we would call it. This is actually torsades, which is oftentimes fatal arrhythmia.
And the Philips system does mark which ones are what they think is ventricular
That's
beats,
right.
but ultimately, like, you know, they do have an algorithm, but a lot of our patients have kind of different looking waveforms because their hearts are different. So it's not always the most accurate. And so a lot of it comes from that.
And that's a totally good point that Jeff's making, that the Philips system really helps us hone in on where the rhythm is abnormal, because as you can see here, it's labeling normal beats and then ventricular beats. So it does call out where there's abnormalities, but there are still limitations in its ability to recognize abnormal versus normal. And so that's why it still requires a manual review of every heartbeat. But that said, it does give us some idea of where the beats are abnormal so that we can hone in on those. I'm going to skip over this because it's not super relevant, but essentially this was just all of our statistics saying that it turns out that the patients that had NSVT or non-sustained ventricular tachycardia are the patients that had their valve placed low in the right ventricular outflow. So if the valve was placed very low, they are more likely to have ventricular tachycardia. If the valve was placed high, they were less likely. So that is a procedural factor that was super important for us, because going forward, now we know we can't choose where the valve wants to go. If it has to sit low because of the configuration of the pulmonary artery, which is shown here, if it has to sit low, which is down here where my mouse is, then that's where it has to sit. But it gives us context that that patient is going to be at more risk. In this particular patient, you can see the valve is this metal structure here. It's actually sitting a little bit higher above the muscle part of the right ventricle. So then we would know, okay, this patient's at less risk for a ventricular tachycardia. So it gives us context and it gives us clues of which patients to be most worried about. And we would not have been able to do that had we not been able to go through and review all of the telemetry. Here's just an example of a patient who had to have the valve placed much lower. So you can see the metal frame is extending much more into down here, which is the right ventricle. And so this patient would be at very high risk for having an event, a ventricular tachycardia event. Here's just an example of blood flow going through the valve. This is a catheter in the right ventricle. The valve is the metal frame there, and then contrast is being injected to show that the valve is seated nicely and working well. But again, this patient has the valve sitting quite low and so would be at risk. I'm going to skip through this, but just onto our clinical management. So this allowed us to devise a clinical algorithm with our electrophysiology colleagues to say which patients we should be particularly concerned about and how we should manage them. So again, without that telemetry data, we would have had a hard time guessing which patients we should be worried about and when we should escalate their monitoring, whether that's moving them to the ICU or escalate their medical therapy, such as starting an antiarrhythmic medication to try to prevent something bad from happening. There are newer valves from other companies. This is the Altera, which looks very similar to the valve that I was just showing you guys. We've done 4 or 5 of these at Packard, and all of them have had ventricular arrhythmia. So again, very helpful to know kind of, it's not the same valve, so we have to think about it slightly differently, but very helpful to know from our other experience that these patients are going to be at risk for ventricular arrhythmia. I'm I'm gonna skip over this just in the interest of time. And we're gonna move on to one of Jeff's other studies.
Yeah, I mean, I think what Anne showed was just really incredible, looking at just the telemetry data and how much even the waveforms can show us. In the interest of time, I'll just go through this really quickly. I think anybody who's in the clinical setting or watches any sort of, drama shows about hospitals, there's a ton of alarms that happen at the bedside. And so actually, like, it's a huge thing that alarms are, a good chunk of alarms are false alarms or nuisance alarms. So sometimes people don't pay attention. It takes away from the real alarms, and it tends to, and there's been a lot of like kind of the Joint Commissions, a lot of different institutions have placed bedside alarms as a serious patient hazard even. And so kind of thinking about how do we improve the bedside experience? How do we, change our alarm parameters? How do we improve the disruption to our families and patients, who have a lot of also anxiety around whenever the alarm goes off? How do we make our alarms more clinically effective for the people that are following them? And so, this is something that we used a lot of, kind of not necessarily the waveform part of it, but a lot of STARR related data lake stuff to download all the alarms that happen in a unit at a time. So this happened on our pediatric ICU as well as our acute care cardiology floors. And so we essentially used the Philips machine to totally revamp how we think about alarms and the timing of alarm delays and everything, to kind of see if we can decrease alarms safely. This is kind of mimicked and adapted from a Cincinnati children's study. And I won't go over this in detail, but essentially, like, we have our mixing lesion patients who are, our hypoxemic children who have a different set of alarms, as well as the ones that are normal hearts that have different saturation. So normally your saturation should be above 90, right? But then just because a baby moves, sometimes the alarm will go off, right? But this allows for more time for the the infant or the patient to self-recover, and then that way we don't we aren't overly sensitive to alarms. And so we'll just go over this really quickly. This is the alarm monitor that's, we know and love clearly. And so, obviously there's going to be a lot of alarms that happen during this time. And so when we did this intervention, all the nurses were educated at that time about how to change all the alarm settings. And this also reflects in a change in kind of the alarm parameters and alarm triggers that go off on the Philips and the vital sign monitoring data download. And so this all occurred back in 2022. So it's 2020 actually. And then so we kind of rolled all these different parameters out, and is adapted from a 2014 study we did here that changed the heart rate parameters. And so we changed the saturation. We didn't change the heart rate, but we changed the respiratory rate, and we changed the kind of ventricular alarm parameters as well. And so just going through this really quickly, at a baseline in 2019 on the PICU, there was 30,000 alarms in the 4-week period. And so that comes out to be about 1,100 alarms a day on a unit, which is it's insane really if you think about it. And then we were able to decrease the total alarms by about 50% there. And then in a follow-up we did about a year ago now, we had sustained effect. And similarly in the acute care cardiology floor, we had 30,000 alarms. So imagine it's like 800 alarms per day. And then we were able to decrease it by 60%. There was a little bit of rebound in the 2-year follow-up, but we can talk about that later. Looking at the alarms per patient, really, you can see how skewed it is. There are certain patients that generate 300-400 alarms a day. And we were kind of able to move the needle a little bit. And here you can see we go from 11 alarms to 2 to 3 in the follow-up and the second follow-up. So really pretty significant 70% alarm decreases over each patient day. And so it really does add up here. And then same story in the acute care in the pediatric ICU, where we went from 12 to 3 and 3 in this era. And so these do include patients who have had 0 alarms. And so those are, we included those patients to kind of tell us when can we have patients that are ready to go home? When can we have patients that are ready to come off alarms, continuous alarms altogether? And so we kind of use that as a metric in the study. There's a lot more details to this that I probably won't go over, but in the interest of time, but just we were able to kind of use the telemetry data, use the alarm data, all through vital sign monitoring, all through this Philips, as well as the STARR data lake to kind of pull all this data and say that, you know, we were able to decrease bedside alarms while not compromising safety. We didn't have increase in code events, increase in deaths, or anything like that. We were able to give a little bit more satisfaction to the nursing staff with more clinically effective and more actionable alarms. Clearly, there's still a lot more to do, and to think about, you know, can we use artificial intelligence? Can we design specific disease-specific alarm parameters, certain customized tools for each patient to kind of further target alarm reduction? I think we're pretty much out of time, so I'll skip this, but I think Anoop talked a little bit about heart rate variability in the ICU. I know there's other NICU, in the neonatal ICU, there's a few people looking at that. There's definitely a lot of challenges in a pediatric ICU, given just that huge age group distribution that really affects our numbers. So it's quite interesting, but I want to think, and so I think the NICU might be a good place to really start. And I won't go over this, but this is some of the data there. I wanted to thank Sanjay, who's helped us a lot with kind of this data waveform monitoring and downloading and going through a lot of these hiccups along the way. So thanks everyone for your attention.
Thank you.
Wow, thank you so much, Jeff and Anne, for such a wonderful content. I'm just looking at time, and perhaps we can allow one question. But I just want to let everybody know that we are just running a little bit behind. So just bear with us and stay on for more exciting content. If there's no question, we can move on to the next speaker. Thank you so much. Our next speaker is Dr. Jennifer Balikai, who practices internal medicine, is also clinical associate professor in primary care and population health, infectious diseases. I've had the pleasure of collaborating with her team on COVID and flu study recruitment. Today, she'll be talking to us on how the real-time alerting is helping her team in effective collaboration in patient recruitment. Jennifer, take it away.
Here we go. Hi, thank you very much, Sanjay, and thank you to the whole research IT team. I see very many familiar names on the list of people here today. Let me see if I can share my screen. I have a brand new computer, which is super exciting, but it told me I wouldn't be able to. Okay, good. So I do not like to be in the middle, like in the way of lunch here. So I'm going to do as quickly as I can. But also highlight, this is one of many chances I had to collaborate with the research IT team. So I won't get to highlight all the other wonderful benefits we had. My role over the last couple of years has been working in a lot of clinical trials related to COVID, and specifically the outpatient studies to prevent severe COVID infection. And one of our challenges, let me see if I can display this. There we go. One of our challenges right off the bat, you know, in 2020, there's a lot of infectious disease specialists with lots of ideas about how to decrease the severity of COVID, maybe with anti-inflammatories, antivirals, what have you. And so a lot of trials got started, right? Everyone's trying to fix the COVID problem. And then all of a sudden we had, you know, at least a dozen trials going at Stanford that were recruiting patients that needed to be sort of seen within 3 days, sometimes 5 days of their diagnosis or their symptom onset. And so the usual STARR tools that we all know and love were not super helpful or useful because of the time delay there. We really needed to have or know who might be eligible, or, you know, connect with people who could potentially be eligible for these research trials sooner rather than later. So that was one of the challenges. And the other important thing to highlight is that we had, you know, dozens of investigators working on these trials, all competing ostensibly for the same group of patients. There were many challenges, a lot of logistical challenges. I don't want to cause any PTSD here, but we were working outside in tents. We were working, you know, we've moved multiple times. We moved to a physical location that we call the COVID CTRU. That's on 835, 845 Page Mill Road. And these are office buildings that were retrofitted and made sure that air quality was and flow was okay for hosting people with COVID in these settings, because the regular 812 CTRU wasn't a safe place with other people that didn't have COVID. And here are all the study teams that were working in all of these various conditions. So these are all the logistical problems that we had, but I feel like the research IT team was like the solid core that helped us like get through a lot of these logistical challenges, because they were very much supporting us in terms of helping us find participants, and specifically for the recruitment challenges. We had to identify people across multiple sites. It wasn't like one clinic that we were going to find people, right? This was a whole population problem. So we had the emergency room, the express care, primary care, and then we had all these COVID testing sites where they weren't even necessarily people who were Stanford patients. We had to identify people within 3 days, sometimes 5 days if we were lucky, of diagnosis. We had to allocate then these participants to multiple different studies, depending on their interest, depending on their eligibility. And then the big one that was really important was to have the optimum number of contacts with these participants. So we had to coordinate across the studies and their study coordinators, so that one person wasn't calling, 5 people weren't calling one person to introduce them to possible studies. So those were sort of our challenges. And I learned about this through Sanjay. So the complex event processing feature that you guys use, is this real-time alerting system. I guess many people or many departments use this, and it's configured to continually read live HL7 feeds. So we can pull people based on their, for example, diagnostic code for a clinic a visit, or we could pull people based on lab values. So let's say the positive COVID test, and it's used in this setting for helping people identify people quickly. So in looking at all the features that CEP has, that's available on some of the published materials that you have, the ones that we really benefited from was the ability to send patient details, not just a name and an MRN, but some other information about their potential eligibility. We could work in conjunction with REDCap, so we could hold this information and utilize it in a workflow setting with multiple different users using different reports. We held this in a relational database, avoiding duplicate alerts for the same person. And then, or actually we could keep one person, we had multiple alerts for one person if they had COVID multiple times, which became important for some of the other studies that we've had. We were also able to integrate it with an external referral system. So we could add referrals that came not just from the CEP alert, but we were able to add referrals from occupational health and other outside groups that we have relationships with. And because of all of this, we thank you, Research IT team. We were able to recruit over 1,600 eligible study participants across these 12 different studies. We had this coordinated outreach to Stanford patients, so people weren't getting annoyed by multiple phone calls. We have this successful system now for capturing outside referrals that we still use. We've had multiple publications, both from investigator initiated here at Stanford and then NIH study publications. You can see some of those here. And then the one I'd like to highlight is that we had a partnership, or we still have a partnership with some of the county clinics where we can focus on recruiting underrepresented populations for these studies, which is a really important part, especially during COVID. So I think I'll stop there. Just the time.
Thank you so much, Jenna, for the wonderful presentation. Especially the last slide, it just very inspiring for all the research technology members, and just makes our jobs seem more worthwhile. So thank you so much. So if there are no questions, I'd like to move to the final speaker before lunch. So Dr. Salamasi is an assistant professor in the Department of Anesthesiology, Perioperative and Pain Medicine. Their clinical practice is focused on neuromodulation and minimally invasive spine procedures. The research focus is pragmatic clinical trials and how to best embed them in clinical practice. So Dr. Salamasi, take it away, please.
Almost good afternoon, everyone. I am the only person between actually you and your lunch break, so I try to make it brief. I have 2 disclaimers to start with. I have not used the STARR as often as I wanted to. I have used that for feasibility of some clinical trial assessment, and also I have used that for some cohort identification, but not a lot of other beautiful features that you showed. I am trying to present more of the things that we do with the big data in our division. And that is my disclaimer number 2, which is a big part of this is the kind of development by my mentor and boss, Dr. Mackey. Initially, he was supposed to give this talk, and he's unfortunately in Scotland. He sent his apologies, and unfortunately, I'm sorry that you're stuck with me. So getting these 2 out of the way, let's start. This is the standard slide that I put at the beginning of every presentation that I have. This is just to show how important my job is as a pain physician and keep me employed. There are about 100 million people in America that have chronic pain, and that is a very costly disease, more than $600 billion, because it is undertreated. We don't have a lot of good evidence for the treatments that we have for these patients either. Now, of course, I'm not going to even talk about the opiate pandemic and all those things at this point. I want to show you some examples of what we have in the field of clinical research in pain. This is one example that is a very good study done by a very reputable pain researcher in Israel, Dr. Eisenberg. They screened about 550 patients, then they only include about 35. The other example that they have is an industry-sponsored clinical trial that led to FDA approval of a device, and they excluded all patients with psychological or psychiatric disorders. That's close to roughly 100% of the patients that I see in my clinic. And that's basically what I'm doing at this point, that looking at the data from these 10 yellow patients that are included in these clinical trials, and then I'm extrapolating that data to these 90 patients that the blue ones that I see in the clinic on a daily basis. And I hope that there is enough similarity between them so that I can just basically start treating them and extrapolating the data from those clinical trials. So that's why we are at this point thinking about having more of supplementing what we have in the traditional explanatory clinical trials. They are fantastic for assessing efficacy. They have very high internal validity if they are designed well. And the quality of the clinical trials are improving, meaning that it is very difficult nowadays to get a good high quality clinical trial into a journal and publish that. So there is usually very high internal validity for most clinical trials. But when it comes to extrapolating the data, generalizing that, that's where a lot of the traditional explanatory clinical trials fall short. That's why if we really want to base our practice on the evidence, we need more of that kind of practice-based evidence, the evidence that comes from our practice to the feedback to our practice as well. So that's why we want to do more pragmatic clinical trials, large comparative effectiveness trials in our practice, and go from one end of this spectrum to the other end of the spectrum, going from just doing explanatory clinical trials and supplementing that by doing more of the pragmatic clinical trials, going from assessing if the intervention works in ideal experimental setting to does that interventional work in real world data, the data that you are generating. And of course, these clinical trials are a little bit different in different aspects of methodology. Inclusion criteria, the way that the intervention is defined, the way that the control is defined. A lot of time the control is not placebo, it's just usual care, and a lot of other things. But all these things is just an arbitrary line. As I showed in that continuum in the slide before, there's no demarcation between pragmatic clinical trials and explanatory clinical trials. They're kind of supplementing each other, complementing each other, and each clinical trial can have features of both ends of the spectrum. So to try to run these pragmatic clinical trials and run them more effectively, we are using a learning healthcare system in our clinic called Choir. Choir is I think the requirement number one, not choir per se, but the learning healthcare system is required number one for doing pragmatic clinical trials. Of course, you need to integrate your clinical trial into medical practice. You need to have a receptive institutional culture, both from my colleagues to help me recruit patients and from the IRB to allow having more flexible consenting and more flexible aspects of even documentation of the consent form. Exactly what Alex was talking about earlier, that there can be challenges without IRB regarding the process of waiving the consent, waiving the documentation of consent. So these are the things that are, I think, evolving. I have worked with IRB for the past 7 years to be able to see how we can streamline the process of consenting the patients for some of these clinical trials. But none of these are possible without having an efficient EHR, an efficient learning health system that can be more dynamic. The one that we have is Choir, and this is something that developed in 2012 by Dr. Mackey, has been in our clinic for the past decade and so. It has some specific questions about the pain experience of the patient, has some promise measures, NIH promise measure, patient-reported outcome measures. And I know that a lot of you are familiar with the promise measures, and you know that to get answers for, say, 12, 13 different promise measures, patients need to answer about 500 to 600 questions. But we use an adaptive testing system so that by 4 to 6 questions per measure, we get to the T-score for the measure and the percentile for the measure. So that's going to decrease the survey burden for the patient. And it is not just a set of questionnaires. There is a big interaction at that intersection of the Choir. It has a good patient interface that I'm going to just show you, the clinician interface that I'm going to show you again as well. And also there are going to be a lot of add-ons to that. And it is really modular, meaning that I can flexibly change that to different ways that I want to collect data for different research projects. And it is free. This is how it looks for a patient. So we have 2 different ways of sending surveys to the patients. Sometimes the surveys are sent based on the point of care. What does it mean? Choir screens the clinic schedule ahead of time and then automatically sends either an email or a text message with a secure link to the patients before the clinic visit. If it is a new patient visit, the initial survey that is longer has some more questions about the background of the patient. If it is a follow-up clinic visit, usually a follow-up survey that has more questions for the promised outcome measures and the trajectory of the pain. And this is one way of collecting data for the patient attached to the point of care, which can be a clinic visit, which can be a procedure. The second way of collecting data is what this question pertains to, the treatment-based survey, meaning that I can start sending surveys after a specific treatment. So I implant a spinal cord stimulator for the patient. That triggers the initiation of a lot of surveys for that patient. These surveys are more targeted towards the treatment. So they ask about patient global impression of the change from that. They ask about adverse events of that intervention, that medication. And also they are timed in a way that are pertinent to that treatment. So for example, if I want to ask questions about a medication, I probably send monthly surveys to the patient. But if I want to ask questions about the spinal cord stimulation, I look at the clinical trials. Most of them report outcome at 1 month, 3 months, 6 months, a year, and further out annually. So my surveys are being sent out to the patient at those same time intervals. And just after the first initial trigger, it is going to continue sending the surveys to the patients. So these are 2 ways that we are collecting data from our patients. And this is the dashboard that I look at when I go to the clinic and I start seeing Vafis Almasi as a patient. And when I click on one of these links, it opens up a PDF report for me that shows this patient's pain distribution, the trend of pain, psychological functioning, physical functioning, sleep, fatigue, and all of those over time. And that's how it helps me manage their clinical care, in addition to, of course, the research-grade data collection. In addition to that, during that point of care, if I have equipoise between treatment A or treatment B, not only me, but every physician in our clinic has the opportunity to randomize the patient to one of these two treatments. And after randomization, this randomization triggers surveys that, again, are treatment-based. So the surveys that are specific to this research study with this randomization are going to be sent to the patient at specific intervals right after this. This is the clinical trial that we concluded, comparing two different medications, duloxetine and cypramine. And again, these are all comers, all patients that come in, and we don't have an equipoise, but we have an equipoise between these two medications. So basically prescribing either or. And the dashboard shows me the randomization arm. I start prescribing the medication, and Choir is going to send them the survey. So I want to take one step back rather than going into details of the studies and showing you the data, which my data is not as exciting as a lot of really beautiful data that I just saw in these presentations. I want to take a step back and show a big picture from a higher altitude. So I start here. I start at the point of care. During this point of care, I either do randomization, I start a treatment, I sometimes do nothing. And then I continue to collect data for these patients, either through routine clinical care or a research study. A lot of people in the field of pain medicine are doing more studies that are focused on finding a biomarker for pain, finding a biomarker for trajectory of pain. Dr. Mackey is focused on doing functional brain imaging for the past couple of decades and trying to see how it predicts pain. In addition to that, there is a big group that is trying to find more specific biomarkers in biomes in genomes to try to see if they can put all of this together into a knowledge network. And I hope that with this knowledge network, I can just continue and apply the medications or the interventions that are more individualized for that patient. And then, of course, rinse and repeat. Do the same data collection, assess if I'm helping that patient, and go continue that. And this is what we are envisioning by using a learning healthcare system, combining that with the STARR Data, combining that with Epic, combining that with a lot of other platforms that we have to try to have a more meaningful data collection and the real world application of our data for these patients. And this is something that is even being promoted by FDA. They say that at this point, approval of a lot of medications, devices is not enough. We need to go one step after that. So if a medication is approved for pain, the number needed to treat is 3.5. It looks great. But what if I start this medication and after 1 year, only 30% of my patients actually continue that medication? Does it still translate to a number needed to treat of 3? I don't think so. The other thing is that all of us know the stories about Vioxx and the other really rare side effects. So that's why the research does not stop after phase 3. The research does not stop after proving efficacy. It continues to real world application. And everything that I saw here was actually that real world application. So I'm excited. Thank you very much, everyone. More than happy to answer any questions.
Thank you, Dr. Salmasi, for the wonderful presentation. And I like how Choir is like at the center of your workflow. So thank you. Thank you once again.
Thank you for having me.
Yeah. So if there are no further questions, I would like to hand it back to Priya. Priya, you're on mute.
Yes. Thanks everybody for joining. That basically concludes our session for today. I hope to see a lot of you back here tomorrow, and tomorrow we start at 1:30 in the afternoon. So have a great rest of your day, and we'll see you tomorrow. Thank you, everyone. Thanks for the presenters, the panelists. Thank you for making this event possible. Thank you.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.